Usually when we talk about a medical diagnosis, there's this expectation of like ultimate precision.
Right. It feels like engineering.
Exactly. It's like you break your arm, the x-ray shows that jagged white line, and the doctor just points at the screen and says, well, there it is. That's the problem.
It's a very binary state. Broken or not broken.
It's clean, it's actionable, and honestly, it's comforting.
Oh, for sure. I mean, human beings like things to be visible. We want our problems to be neatly categorized so they can be, neatly fixed.
But then you step into the world of oncology, specifically blood cancers, and suddenly that x-ray machine is just utterly useless.
It really is.
And we are looking at a therapeutic landscape right now that presents an incredible, almost cruel paradox.
Yeah.
If a patient comes in with a B-cell cancer, like certain types of leukemia,
–
we have built absolute medical miracles for them.
Yeah.
I mean, targeted therapies that act like a smart bomb.
Right. They just sweep through the body and wipe out every single B cell to guarantee the cancer is eradicated.
And the patient survives that.
Mm.
But flip the coin. If a patient relapses with a T-cell cancer, they're left staring at a dismal 5-year survival rate of just 7 to 38%.
Which is staggering.
It is. Why is there this massive difference?
Well, it really comes down to a terrifying biological roadblock. You see, the human body is surprisingly resilient when it comes to B cells.
Right.
We can actually tolerate the temporary depletion of the entire B-cell population. We wipe the slate clean, kill the cancer, and keep the patient safe with supportive care while their B-cells eventually grow back.
But T-cells are entirely different.
Entirely different. T-cells are the generals of your immune system.
Right.
So if you tried that exact same pan-depletion strategy, just wiping out all the T-cells to ensure you kill the P-cell cancer, you trigger profound, fatal immunosuppression.
The patient basically becomes completely defenseless.
Exactly. Defenseless against even the mildest everyday pathogens.
So that is the puzzle we are unpacking today. How do you assassinate the cancerous T-cells, but spare exactly enough normal T-cells to keep the patient alive?
It's a massive challenge.
It is. And the mission of this deep dive is to explore a groundbreaking paper recently published in Nature Cancer.
A really fantastic paper.
Yeah. We are going to examine the creation of a microscopic sniper-like therapy engineered specifically for T-cell cancers. We're going to pull apart the mechanisms, the biological tricks, and the sheer cleverness these researchers used to solve a problem that seemed impossible.
So we should probably start with the biology of the target itself, right? The T-cell receptor, or TCR.
Yes, let's start there, because to build a sniper, you need an incredibly precise target.
Exactly. The researchers focused on something called the alpha-beta T-cell receptor. Now, this receptor is present on the surface of over 90% of cutaneous T-cell lymphomas.
The CTCLs.
Right, and about 30 to 50% of T-cell acute lymphoblastic leukemias, or TALs.
So it's a huge target.
It is. And creating this receptor during a normal T-cell's development is essentially a game of genetic roulette.
I love that analogy.
It undergoes a process called somatic recombination. The cell basically shuffles different genetic segments to build a unique receptor.
And those segments are the variable region, the diversity region, the joining region, and finally a constant region, right?
You got it. V, D, J, and C. It uses these to build a receptor that can recognize specific threats.
I want to zoom in on that last piece, the constant region, or the TRBC segment, because this is where the massive vulnerability lies for the cancer.
Oh, absolutely.
When a T-cell is building its receptor, it has to choose between two available genetic alleles for this constant region, TRBC1 or TRBC2.
Right. It cannot have both.
It has to pick one.
And that mutually exclusive choice is the foundation of this entire therapy. In a healthy person, your normal T cells are a beautifully diverse mix.
Right.
By pure random chance, about 40% of your T cells selected the TRBC1 allele, and the remaining 60% selected TRBC2.
It's a blended, balanced population.
Exactly.
I like to picture this as like the left-handed versus right-handed army analogy.
Oh, that's a good one.
Right. Imagine your healthy immune system is a vast army of soldiers. 40% of your soldiers hold their swords in their left hand, that's TRBC1,
Okay.
and 60% hold it in their right hand, that's TRBC2. But cancer doesn't play by the rules of a normal, diverse army.
No, it doesn't. Cancer is a clonal army.
Right. It starts when one single rogue soldier goes mad and just copies themselves millions and millions of times.
Precisely. Because all the cancer cells are exact genetic clones of that one original rogue cell, a patient's entire T-cell cancer will be exclusively TRBC1 positive or exclusively TRBC2 positive.
It will never be a mix.
Never.
So the strategy just writes itself. If a patient has a TRBC2 positive cancer, you don't drop a bomb on the whole immune system.
You design a targeted weapon that only recognizes the right-handed soldiers.
Yes, the TRBC2 cells. You wipe out 100% of the cancer, but you completely ignore and spare the 40% of normal T cells that are TRBC1 positive.
And preserving that 40% of normal T cells is the theoretical key. It leaves the patient with enough immune function to stay alive.
Avoiding that fatal immunosuppression we talked about earlier.
Yeah.
Okay, so let's do a quick recap here to lock this all in before we move on.
Sounds good.
Bullet 1, B-cell therapies wipe out everything safely, but T-cell therapies can't because you die of infection.
Right.
Bullet 2. T-cell receptors have constant regions, and they have to choose either TRBC1 or TRBC2.
Yep.
And bullet 3. Cancers are clones, so they are stuck with just one TRBC allele.
Perfect summary.
So here is a question for you, listening at home. Imagine your immune system as a deck of red and black cards. If a rogue black card copies itself 1,000,000 times, how do you burn the rogue cards without touching the red ones?
That is the exact engineering problem the researchers faced.
Right, which makes you wonder, if you have a rogue clone copying itself 1,000,000 times, how do you engineer a drug that only recognizes one incredibly specific microscopic feature?
Because it's not like they didn't try before.
Exactly. The paper mentions we already had tools that tried to do this. There were existing antibodies out there, named YR3A5, KFN, and SAMBOC2, that supposedly targeted TRBC2.
They did exist, yeah.
So why couldn't the researchers just use those off-the-shelf parts?
Well, because in the world of molecular targeting, off-the-shelf parts are often too weak or, honestly, too sloppy.
Sloppy how?
When we evaluate how well an antibody grabs onto its target, we look at affinity. We measure this using something called the dissociation constant, or KD.
Okay, KD.
Yeah. They measure this using surface plasmon resonance, or SPR.
What is that, practically speaking?
You can think of SPR like a microscopic wind tunnel. It tests exactly how tightly two molecules bind to each other under physical stress.
Oh, wow. And the results for the old antibodies weren't great.
No, they were totally inadequate. For a highly effective clinical cancer drug, you want a tight, strong grip. But the existing KFN antibody had incredibly weak affinity.
It had a high KD, right? Like 175 nanomolar.
Exactly. It would just fall off the cancer cell. And the SAM.2 antibody had a much more dangerous problem, cross-reactivity.
Meaning it hit the wrong targets.
Yeah. It bound to the target TRBC2, sure, but it also accidentally bound to TRBC1.
And if you use a sloppy antibody that accidentally hits TRBC1, you end up killing the normal, healthy T cells we are desperately trying to save.
It defeats the entire purpose.
Right.
So they needed to build a fundamentally better magnet. They started with a YR3A5 antibody and subjected it to a technique called phage display.
Okay, phage display. This is where the biological engineering becomes incredibly elegant.
It really is. And an antibody grabs its target using these microscopic grasping fingers called complementarity determining regions, or CDRs.
There's 6 of these regions, right?
Yep, 6 CDRs.
What?
The researchers took those grasping fingers and systematically mutated them. They swapped out the amino acids for all 19 other variations, generating 1,406 unique viral clones, or phages.
Wow.
Each one was wearing a slightly mutated unique antibody on its surface.
It's essentially an evolutionary brute force attack. You're trying 1,000 different microscopic keys in a lock to see which one turns the smoothest.
That's a great way to look at it. And to figure out which of those 1,400 mutants was the absolute best, they used a custom sorting algorithm and assay called SLICY.
SLISY, which stands for Sequencing Linked Immunosorbent Assay.
Right. They washed all these mutated phages over TRBC2 cancer cells to see what stuck.
But crucially, they also watched them over TRBC1 cells to check for that dangerous sloppiness.
Exactly. Then, using massively parallel sequencing, or MPS, they could literally read the genetic code of the winners.
They were calculating binding ratios and enrichment ratios. Basically, if a mutant stuck like glue to the cancer cells completely ignored the healthy TRBC1 cells, its genetic sequence lit up in the data.
And through this massive screening, a clear winner emerged. A mutated antibody they named JX1.1.
JX1.1.
Yeah. It had 3 very specific structural tweaks, amino acid substitutions. Specifically, mutations V30K, S32R, and R27Y.
Just those 3 tiny changes.
Just those 3. But they changed everything. It gave JX1.1 a staggering affinity. But what's really fascinating is the mechanics of its slow off rate.
Okay, the off rate.
Think of the off rate as how long the antibody refuses to let go once it grabs on.
Like a bulldog locking its jaw.
Exactly like that. And the math bears this out beautifully. They calculate this using a formula, e to the power of negative kd times time.
Okay, so if we run that math.
Because of its specific structural tweaks, the off rate, or lowercase kd, was just 0.0013 inverse seconds, meaning if you wait 10 minutes, about 45.7% of the new JX1.1 antibody is still firmly clamped onto the target cell.
Wow, and if you look at the old YR3A5 antibody.
Less than 15% was left holding on. JX1.1 simply does not let go.
Okay, wait, I have to jump in and play devil's advocate here.
Go for it.
We built a better magnet. We proved it clamps on like a bulldog. JX1.1 sticks to the outside of a TRBC2 cancer cell perfectly. But simply sticking to the outside of a cell doesn't kill it. If the drug is just sitting there on the surface, how does the actual assassination happen?
That is the crucial next step. JX1.1 on its own is not a weapon.
Right.
It is purely a navigation and delivery vehicle. To kill the cancer, we have to turn it into an antibody-drug conjugate, or ADC.
Okay, time for our next 5-minute recap before we get into the ADC.
Let's do it.
Bullet 1, old antibodies were just too weak or too sloppy.
Way too sloppy.
Bullet 2, phage display and the SLIEY assay allowed them to test thousands of mutants.
Yeah.
And bullet 3, JX1.1 emerged with an incredible grip and a super slow off rate.
Spot on.
So, my question for you listening, think about Velcro versus superglue. If a drug's effectiveness depends on how long it stays stuck to a cell, how does that change the way we design medicines?
It changes everything.
It really does. So to recap where we are biologically, we engineered an incredibly sticky antibody called JX1.1 that perfectly targets the cancer and ignores healthy cells. But because sticking to the outside isn't enough, we have to weaponize it. Which brings up an amazing biological mechanism. If an ADC relies on the cell swallowing the drug, how do we prove that actually happens?
The researchers did this visually, and it's brilliant. They attached a specialized dye called Frodo to the JX1.1 antibody.
Frodo.
Yeah. Now, Frodo is a really clever chemical. It's completely invisible when it's in a neutral pH environment, like floating in the bloodstream.
Okay.
But if it enters a highly acidic environment, it suddenly glows bright red.
And that's exactly what they wanted to test.
Exactly. When the JX1.1 antibody binds to the T-cell receptor on the surface of the cancer cell, the cell does what cells naturally do. It recycles its surface receptors.
Right, it pulls the receptor inside, dragging the attached antibody with it.
Yep, it drags it into an internal bubble called an endosome. That endosome then travels deeper into the cell and fuses with a lysosome.
Which is essentially the cell's garbage disposal or stomach.
Okay.
And the inside of a lysosome is highly acidic.
Very acidic. So under the microscope, the researchers were able to watch these living cancer cells slowly light up with bright red fluorescence from the inside out.
Wow.
It was absolute visual proof that the Trojan horse had been pulled through the gates, past the cell wall, and straight into the acidic stomach of the cell.
That is so cool. And once it's inside that acidic lysosome, the real assassination happened.
Yeah.
Because the researchers had conjugated or linked the antibody to a warhead called SG3249.
Right, which is a paralobenzodiazepine, or PBD dimer.
PDD dimer. What's brilliant is how it's attached. It uses a cleavable linker.
Yes.
It's a chemical bond explicitly designed to hold together tightly in the neutral bloodstream, but the absolute second it hits the acid of the lysosome, it snaps.
The acid hits, the linker shatters, and the poison is unleashed. The PBD dimer escapes the lysosome, enters the cell's nucleus, and chemically cross-links the cancer cell's DNA.
Cross-links it.
It essentially stitches the double helix together so tightly that the DNA cannot unzip to replicate.
And a cell needs to unzip its DNA to survive.
Exactly. When a cell realizes its DNA is irreparably jammed, it triggers apoptosis.
Cellular suicide.
Right. And the researchers engineered this so efficiently that they achieved a drug-to-antibody ratio, or DR, of about 4.3 to 4.9.
Meaning every single guided missile is carrying between 4 and 5 individual warheads of this lethal poison directly into the cell's nucleus.
It's an overwhelmingly lethal payload delivered with pinpoint accuracy.
So if we pause and look at this Trojan horse mechanism, it's staggering. Let's recap this section.
Okay.
Bullet 1. ADCs use antibodies as Trojan horses.
Yep.
Bullet 2. The TCR swallows the antibody into an acidic lysosome.
Right.
And bullet 3. The acid releases a DNA cross-linking payload, destroying the cell from within.
A perfect assassination.
So, question for the audience. If cancer cells naturally recycle their surface receptors, what other cellular garbage disposal systems could we hijack to sneak medicine inside?
It opens up so many possibilities.
It really does.
Yeah.
But of course, making a guided missile work in a test tube is one thing. Does this actually save lives? Let's look at the graded clinical evidence, because this is where the theory hits reality.
We have to move through the evidence tiers carefully. First, in vitro in the Petri dish.
The lab results.
Exactly. The JX1.1 ADC showed massive cytotoxicity against TRBC2 positive cancer cells.
They measured an IC50 of 0.3 to 1.8 nanograms per milliliter, right?
Yes, which is highly potent. It's killing the cancer at concentrations perfectly in line with successful clinical B-cell therapies.
Wow.
And crucially, when they mixed healthy and cancerous cells together in the dish, the normal TRBC1 cells were completely ignored. They survived untouched.
But humans aren't petri dishes.
No, they are not.
So the researchers moved up a tier to patient ex vivo data. They took blood samples from 3 actual human beings suffering from cutaneous T-cell lymphoma.
Actual CTCL patients.
Now, if you're looking at a microscopic slide of a patient's blood, how do you even know which T-cells are the cancerous ones and which are the healthy bystanders?
That's a great question. The researchers relied on a surface marker called CD26.
CD26.
Right. In these specific patients, the malignant cancer clone had mutated and lost the CD26 marker. So they knew that any cell that was TRBC2 positive but CD26 negative was a confirmed cancer cell.
That makes sense. And the real-world variety between these patients was stark.
Very.
Patient 2 had a moderate tumor burden. About 22% of their T-cells were malignant. But patient 3, it was devastating.
Yeah, a staggering 98% of their circulating T cells were the cancer clone. Just a massive, overwhelming disease burden.
But when they applied the JX1.1 ADC to these human blood samples, what happened?
The results were surgical. It selectively slaughtered the TRBC2 positive CD26 negative cancer cells in both patients.
Entirely, regardless of whether the burden was 22% or 98%.
Right, while beautifully sparing the normal TRBC1 cells.
Which gave them the definitive confidence to move to in vivo evidence-live animal models.
Yes. They used severely immunodeficient mice, called NSG mice, and injected them with TRBC2 positive cancer cells.
But they genetically engineered those cancer cells to express luciferase and green fluorescent protein.
GFP, meaning the cancer literally glowed in the dark.
Allowing the researchers to track its spread inside the living mouse under a specialized camera.
Exactly. In the control group, the mice given a dummy therapy, the imaging showed the cancer rapidly expanding throughout their bodies.
It's aggressive.
Very. Those mice suffered weight loss, hind leg paralysis as the cancer invaded their nervous systems, and ultimately, death.
But the mice treated with the JX1.1 ADC.
Complete clearance. No glowing cancer expansion on the imaging, no weight loss, no paralysis. They survived.
That is incredible.
And this wasn't a fluke. It was replicated again in patient-derived xenografts, or PDX models, where actual pieces of human tumors are grown in the mice.
And the ADC eradicated the circulating tumor cells there as well.
It's the ultimate validation of the mechanism.
So let's lock in this proof with another recap.
Okay.
Bullet 1. The ADC works in a petri dish.
Yep.
Bullet 2, it works in human blood samples from actual lymphoma patients, regardless of tumor burden.
Right.
And bullet 3, it prevents paralysis and death in live mouse models.
It really hits all the tiers.
So for the listener, we've moved from petri dishes to human blood to mouse models. When you look at clinical data, what kind of evidence makes you finally say, okay, this is real?
It's a tough bar to clear.
It is. Because for me, seeing a live animal avoid nervous system paralysis and survive is the emotional tipping point.
Oh, absolutely.
But as amazing as this sounds, I have to step back and look at the broader picture here. Let's talk about the safety.
We have to.
Even with this incredible precision, we are still intentionally wiping out 60% of a patient's normal T cells. Isn't that inherently dangerous?
It is the most critical question we can ask. We have to run the clinical math here.
Let's run it.
A healthy human has a normal CD4 T cell count of between 500 and 1,500 cells per microliter of blood.
Okay, 500 to 1,500.
Right. We know from decades of studying HIV and immunodeficiency that severe, life-threatening, opportunistic infections typically only happen when a patient's CD4 count drops below 200.
Okay, so 200 is the red line.
Yes. If a patient receives this TRBC2 therapy, we expect to deplete that 60% of their T cells.
Right.
Mathematically, based on baseline averages, they would retain between 200 and 360 CD4 cells and 60 to 400 CD8 cells.
Wow. That is right on the edge of the cliff.
It is close to the line, without a doubt. It is mathematically viable, meaning it is enough to survive and maintain a baseline cellular immunity, but these patients would absolutely require close monitoring.
I would imagine so.
Some might temporarily need antimicrobial prophylaxis just to prevent infections while their body adjusts.
And then there's the payload itself to worry about.
Exactly. The PBD dimer is a potent chemotherapy, and it carries standard toxicities, things like cytopenia, which is a dangerous drop in blood counts, or elevated liver enzymes.
It's a heavy hammer. Are there alternatives that don't wipe out 60% of the immune system? What about targeting the variable region instead of the constant region?
You could theoretically target the variable region of the receptor, the TRBV segment.
The V part of the VDJC.
Right, because there are over 30 different TRBV families. Targeting just one specific family would preserve 95% or more of the normal T cells.
That sounds much safer.
It does, but the problem is pure logistics. You would have to engineer, thoroughly test, and manufacture over 30 distinct, highly complex ADCs to ensure you had a drug for every possible patient.
Oh, that would be a nightmare for manufacturing and clinical trials.
Totally unfeasible. Targeting the TRBC region, where there are only two options, is infinitely more practical for creating an off-the-shelf clinical drug that can be deployed quickly.
Okay, but here is where this paper drops an absolute bombshell.
I love this part.
We've been talking about blood cancer this entire time, but at the very end of this research, they introduce a massive Easter egg regarding an entirely different field of medicine, autoimmunity.
This is where we connect the specific mechanism to the broader picture.
Right.
Many severe autoimmune diseases, conditions like celiac disease, rheumatoid arthritis, vitiligo, or graft versus host disease, are driven by what we call oligoclonal self-reactive T cells.
Okay, break that down.
This means a very small, rogue population of your own T cells gets confused and decides to attack your own joints or your own skin.
And just like the cancer clones we've been discussing, if that small, rogue population of arthritis-causing T cells happens to share the same TRBC segment,
you could use this exact same sniper rifle to take them out.
that is just mind-blowing.
It is. If we map the TRBC status of those self-reactive clones causing the arthritis or the vitiligo, this incredibly lethal cancer therapy could be entirely repurposed.
Wow.
It could fundamentally alter how we treat severe, debilitating autoimmune disorders.
Okay, final recap time.
Let's hear it.
Bullet 1. Surviving on 40% of your T cells is mathematically viable, but requires serious monitoring.
Very true.
Bullet 2. Payload toxicity is a standard risk with these heavy chemotherapies.
Yep.
And bullet 3. This technology isn't just for cancer. It could absolutely revolutionize autoimmune treatments.
It's a paradigm shift.
So my final question for you listening. If a treatment is mathematically safe but sits that dangerously close to the edge of immunodeficiency, most people would take the risk to cure a terminal cancer. But would you take that same risk to cure severe arthritis? Where is the line between a necessary risk and an unacceptable one?
It raises profound questions about risk versus reward, especially as our targeted therapies become infinitely more aggressive and capable.
It really does. You know, we spend so much time in medicine putting things into neat little boxes.
You do.
The oncology ward is on the 4th floor. The rheumatology clinic is on the 2nd floor. We treat them like entirely different worlds. But what if the ultimate cure for your body, attacking your own joints, is the exact same Trojan horse designed to assassinate a blood cancer?
It completely blurs the lines.
Think about the massive possibilities if we start mapping the TRBC status of every autoimmune disorder in the world. Suddenly, that broken X-ray machine we talked about at the very beginning of this deep dive,
Yeah.
it doesn't look so broken anymore. It just looks like we finally learned how to read a completely different kind of film.
Beautifully said.
Thank you for joining us as we unpacked this incredible science. Keep exploring the microscopic battles happening inside you every single day.